Thank you so much, Sherry. It's really a pleasure to be with you today. And I get to really welcome you to this onboarding event as we talk about really this exciting time of having the platform live and getting data out to researchers, to make a difference. We are building a platform, not a research project. And we really need you to come in and do research on this. You can go ahead and go to the next slide. Thank you for joining us today. And I'm really excited with the experience that we have. And so, as we thought about this project and really creating a transformational resource, originally with the name, a very catchy name, the Precision Medicine Initiative Cohort Program, which got renamed the much more apt All of Us Research Program, which gives us both an aspiration and a goal to strive towards. We thought of how longitudinal cohorts historically had so transformed disease treatment. And these come in many forms. I give you just one example here from Framingham, which began in 1948 and published its first paper 13 years later. This is a shot from that paper. And really started to tell us that things like cholesterols of 300 and systolic blood pressures of 180, were actually not just part of normal aging, but intervenable risk factors. And if we stop smoking and exercise more and these kinds of things, that we could actually dramatically change cardiovascular mortality. You can go ahead and advance. And what we see over the next 40 years were, just a dramatic decrease in cardiovascular disease, really informed by the intervenable risk factors elucidated by programs such as these. And so, next slide. What we want to do is think about how we can transform the next 40 years of medicine. How do we, create that foundation that lets us discover and those next intervenable risk factors and layer upon our knowledge the dense data that omic kind of data that we can derive. Next slide. And so, but one of the things that's really been a challenge for this is the lack of diversity in many prior research studies. And this is maybe most pronounced and most easily measured within genomics, but it really is across, our clinical trials. It's across a lot of our observational cohorts. And so that's a really a calling card for, what we need to do, and really a scientific imperative, as well as what we could consider an ethical imperative for our program. And so you can see the distribution of genetic studies by GWAS studies, mostly European ancestry. And about 4% of all GWAS today, cover about a third of the U.S. population. So that's really an opportunity for our country and our program to diversify that, and that has impacts upon how we interpret genetic variants. Knowledge and discoveries like PCSK9, which are the most recent discovery in terms of countering hyperlipidemia, and has dramatic effects on heart disease, discovered in a small population of African Americans from Dallas heart study. And then creating things like polygenic risk scores, really work in the populations for which they're derived. We need diverse populations to make those accurate. We need diverse populations to interpret genetic variants accurately. And all these things impact all of us. Not just the All of Us research study, but they impact every single individual and really will help create a global platform for discovery and scientific interpretation. Next slide. So as we thought about the program and designed it, we really want to capture environment, lifestyle, and biology. We want to think about the influence and of these different things. And we want to assess this through, not just surveys and in-person measures, but the robustness of electronic health records, really a place where I've spent most of my research career, and countering that and crossing that with things like molecular data, genomics being a key part of that, mobile, wearable, digital health technology kinds of data. And in this process, we really want to respect participants as partners. We've set the goal of 1 million people to come in and partner with us, and to reflect the diversity of the United States. And I've shared the 2 URLs here. You'll hear a whole lot more about researchallofus.org through the course of this meeting. I'm going to hit on it very briefly. Kelsey will get in more deeply, and then the presentations will talk about the research in it. And then joinallofus.org is where you can join to be a participant. anyone can join. I encourage everyone on this call who's not already a member to join as I have. All right, next slide. So as we think about our program, we launched nationally in 2018. You can see the growth of the program. The lower curve there are people who have contributed biospecimens, have agreed to share electronic health records, and have completed physical measures. And you can see, like many such programs, we were impacted by COVID-19, and really everyone in science has in some way pivoted with COVID-19. And that meant a pause of our in-person activities, which we have since restarted and are starting to scale and open back those clinics back up to re-enroll those in-person elements. But currently we stand at 361,000 people who have signed the consent, and 230,000 of those have electronic health records. 277,000 people have donated biospecimens. Next slide. So this just gives you a sense of our breakdown geographically by age and race and ethnicity. Over 80% of our core participants are underrepresented biomedical research, which includes a number of different categories, not just race and ethnicity, but also socioeconomic status, rural location, sexual gender minorities, and other conditions. And you can see that over half of our population are diverse by race and ethnicity. Next slide. So the major elements of the protocol are, of course, enrollment and consent and EHR authorization. At current time, you have to be over 18 or older to consent. It's an online platform which allows anyone to enroll across the United States. We do plan to expand to children later. Next. And then we collect surveys, physical measures, and biospecimens. The physical measures and biospecimens require an in-person visit. At this point, although right now we are starting and trialing successfully saliva for biosamples that can be mailed to your home. And we look to be expanding that, especially in terms of where we are right now with COVID, and other ways of collecting biosamples without having to actually physically come into a large academic health center, for instance. The surveys cover a number of topics, and recently we released one on COVID, which we'll talk about more through this talk. And then next, mobile and wearable technologies. For that, we have currently allowed people to share their Fitbit and Apple Health Kit data, and we'll be looking to engage other technologies in that front over time. Next slide. So, a big part of this program is return of results, and participants told us through engagement studios and surveys that return of genetic results is really one of the things that they are most interested in. So if you, next, one of the first things we're going to be returning are genetic ancestry and traits, and those data are generated by our genome partners, and from genotyping arrays, we'll be returning those. We're actually in a very small scale piloting that right now, and we hope to release this really over the next month. This should be coming out in a more public way. And then the other thing that we really are excited to return are health-related genetics next on this. And our genetics here will be driven by whole genome sequencing, which we intend to do in 1 million people. And we're initially starting with the so-called ACMG59 genes, which are actionable genes for things like inherited cardiomyopathies, arrhythmias, cancer syndromes, for which there's something that can be done if you find a genetic variant. And then we're also, we'll be returning pharmacogenetic information, and we hope to return this sometime in the next year or so. These informations are supported by a genetic counseling resource as well, so that participants, as they receive this information, or even any sort of genetic information they have, they can get more information about the results and counseling around what this may mean for them. Next slide. So what does some of this look like? So these are some screenshots of our interface for returning genetic ancestry and traits. Initially, we just have those 4 traits there, and you can see what it looks like for cilantro preference. There's an option at the end to give some feedback, as well as if if we predicted it correctly. And then this is the kind of thing that will grow over time. Next slide. And this is a slide of the genetic ancestry. This is a very important topic, and I'll say that we spent a lot of time engaging with different points of participants and participant review process to really try to get this right. It's whether or not you taste cilantro is probably not people don't have really strong opinions on that. They have strong opinions about eating cilantro, but maybe not whether the genetics predict you taste it. But the ancestry component is something that is we wanted to do with sensitivity, and we went through a lot of iterations of the pictures we put through in the text, and hopefully we've done this in a thoughtful way that is sensitive to where we are as a country and are right now. Next slide. So as we think about the scientific framework, this can be used for all sorts of things. I'm really excited to see what you as researchers discover that we haven't thought of. But some of the core things, and we want to have a scientific underpinning, of course, for any cohort of research and prevention, and how do you better diagnose and treat conditions, and what are the outcomes? How can you predict prognosis? But something we think will really be helpful for is thinking about health equity. And about wellness and resilience, because we're not selecting for any disease population, we're selecting in a neutral way. Next slide. So those are our scientific underpinning. And to fill this, we have a number of different tiers of data. And Kelsey's going to walk through this more, and you'll see examples, especially driven on what that first individual research tier that we have out there called the register tier. And there's also a public tier that anyone can go explore. No login needed. You can get counts on many different conditions through the data browser. And then through the researcher workbench, you can go dive into the individual row level data on participants. All of our data will have the obvious identifiers removed from it. Names, social security numbers, medical record numbers, all that kind of stuff is removed. And then the more protected tier is that what's currently available called the registered tier. And over time, we hope to release a lot more information in what we call a control tier. That's not out yet. That will be coming over time. And then eventually, we want to create technologies for people to recontact participants as well, and use biospecimens outside the central use that we're driving as a program. Those are things we're thinking about and will develop over time. Not ready yet. Next slide. So, we, by virtue of our electronic health record data, have many decades, and sometimes data for many decades, sometimes before a participant actually enrolled. That middle row for our surveys, of course, they had to be an enrollee to come in and share survey data. But you can see we have just millions of diagnoses and procedures and drugs that are mentioned in people's charts, and lab and measurement data that are essentially available for use free, for which you can use in a prospective fashion back in time. Next slide. And I just wanted to give an example of a little bit of screenshot of the Research Hub here and the data browser, where you can explore the condition that you can access without logging in, of the key elements. Next. And then we have the Researcher Workbench, where you have to apply to get access. Hopefully many of you probably have already done this, and many of you, if you haven't, will be intrigued and enough to go do this. Next slide. And so just to give you one example, you're going to hear a bunch of examples through the course of this webinar. This was a project looking at depression medication sequencing. So the idea is define who has depression, which you do from electronic health records in this case, and then look at what their first medication is, their second medication, et cetera. And so that inner ring is their first medication. And we just broke this up very simply as white and non-white. You can imagine many more ways to divide this. The purpose of this study wasn't to discover anything new, but nonetheless, we see an interesting difference that I want to note. So you can see for both populations, SSRIs are the first medication used, but you can see the second, third, fourth medications differ between these two populations. That could be because of a whole host of reasons. That could represent something biological too, as well as, in response, that could represent differences in geography, that could represent all sorts of things. What I'm showing is we have the power to look at the sequence of the medication and start to look at some of the drivers and differences in populations. And these kinds of things highlight a research use case. Next slide. So as the rest of the world hit, we have adjusted for COVID-19 as well, and wanted to look at how all of us could help with COVID-19 next. So we looked at our different components, first with surveys, next. We designed a survey called the COVID Participant Experience Survey, or COPE survey, and we wanted to go beyond just the common factors of, did you have COVID, what happened, but we also asked things about how it changed your life, and how it influenced you. So did you social distance? Currently, how do you feel about a vaccine? We asked questions about discrimination, optimism, resilience type questions in there as well, and financial impact of COVID. Next. With biospecimens, we realized that we had a geographic sample from across the country from, in the early stages of this pandemic. So we have taken samples from January through March, really before there were, many known cases in the US, and we've tested for antibodies. We've had a pretty rigorous design with our antibody testing. We're actually employing at some stage 4 different antibody tests against 2 different epitopes. And we're looking at how these relay and hope to add to the general knowledge of serology testing, as well as where and when the people were diagnosed, what the prevalence was of COVID-19 early in the pandemic's hit in the US. Next. And then the next thing would be the EHR data. This may seem obvious, but the EHR data wasn't built for COVID-19 until it hit. So we wanted to work with our many data providers to have the standards of data implemented so that it will be usable by researchers. And we've actually partnered with a number of other efforts, and some of the people on this call may be partnered with some of those other efforts across the NIH as what these data standards look like. I look forward to making these kinds of data in the future available to researchers as well. Next. So where are we now and what will we have? So our current data set, which you can access, includes 224,000 participants, and with survey data and physical measurement data, you can see the amount of EHR data. Very soon, and Kelsey will tell you more about this, we're going to release a larger data set, which will include more information and more participants, but will also include some Fitbit data. We'll also include some of the COPE survey data, so that initial COVID data. And at some point, not directly with this release, but at some point we'll also have a release with our serology data that will get out there as soon as that's ready. And then later, in about a year, we'll have another release, which will be where we'll get at something I know that many of you are probably interested in, which is genomics. We anticipate our first genomics release probably happening in about a year. We're just scaling up that pipeline now. Everything looks good with it, and how we're, how we've designed it, and looking to get enough volume through there that we'll be able to have a significant release in genomics probably in about a year, as well as getting out, the COVID EHR data, the rest of the survey data, and more participants as always. So with that, I'll certainly thank you for joining. I really appreciate your attention and everyone that's here. And I look forward to the rest of the presentation. And I hope that you'll hang around as well. Sherry, I'll turn it over to you to moderate for if there are any questions.
Thank you so much, Josh. Yes, the slides will be available after this session is over for all of the presenters, and we will be recording the sessions as well. So you'll actually get to hear the voices as well as see the slides. If you have any open questions, feel free to put them in the Q&A session of the Zoom. I know we have a few. Josh, someone's asked, what types of EHR do we have available? Is it just Epic or many others?
It's many. So we were just looking at this recently. It's at least 10 different EHR vendors, and of course, many different versions of those EHRs, and many sites have actually gone through migrations. All the data are taken from the different vendors and mapped to the OMOP data model, common data model, and then uploaded. And there are a couple different QC quality control processes in those steps. And so as you come in, you access it through one common data model. They've all been harmonized across their different sources. And it's about upwards of 40 different sites actually uploading data. And some of those are, represent aggregations in and of themselves.
Okay. Another question about the EHR is what prescription information is available and how do we get that?
Yeah, so it's the prescription data that would be part of the EHR. So if a particular entity is an outpatient only entity, so just be outpatient electronic prescribing entries at that point. If they have both inpatient and outpatient, it would have both. So most of our large providers are going to have both inpatient and outpatient prescription data. We also would love and have explored, though have no definitive plans yet, on how we could link in claims kind of prescription data as well. So right now it's just coming from the EHRs.
Okay. One last question from the EHR is, do we have radiology images and clinical notes?
We do not yet, but it's also those are also things on the roadmap. So our desire is to make this a holistic EHR capture, including narrative content, and then eventually we would love to also capture images. Certainly we're on the path to capture narrative content. How we actually make that available, you think in a de-identified way, is, it's challenging, right? To really assert that these are de-identified and don't have names and things like that embedded in them. But there are good ways and approaches we have for dealing with that. And I'll just emphasize that narrative content is much bigger than just, clinical notes. It's also, procedure reports, path and imaging reports, things like that. And some of those may be actually some of the things we get out there earlier.
Can you give us an idea of your plans for genomics? How many people will be sequenced through whole genome sequencing? And then what are our plans for methylation, proteomics, and other omics?
Sure. So our plans are to currently do both genotyping arrays and whole genome sequencing on 1 million individuals. We're doing both because one proceeds faster than the other, and that's at least in our current plans. And then in terms of other omics down the road, those are all things we've talked about. We don't have any definitive plans with that yet. Just like we've actually also done a long read sequencing pilot, or I should say we have plans to do a long read sequencing pilot that will likely be happening within 2021. Those data may take longer to come out. But we will be designing, investigating many other omic technologies over time.
Can you give us an idea of what physical activity data will be collected and specifically how Fitbit data is being used currently?
Oh, those are great questions. So, this is an area where you can capture information from multiple in different avenues. So, there's actually physical activity data that's part of our current COPE survey. There would be, we've looked at future surveys that capture some of that with standardized instruments. And then you have things like Fitbit data right now and Apple Health Kit data that people are just linking in their, their own personal devices. And that, brings to it whatever data that those are capturing. So, people are linking in different kinds of devices. So some would have heart rate, some would not, for instance. Probably everybody has steps coming in from those different providers. And then we will also look at doing some things centrally, probably not necessarily on all 1 million, but different populations. We've already designed a pilot or in the stages of designing a Fitbit pilot that would deliver Fitbit devices to a subset of our population to capture that in a defined way. So, so we look at capturing activity data in multiple different formats. In the ones we collect will be, of course, standardized, but we're also, the whole bring your own device thing will allow us, these devices to become more and more popular, a richer data set that you can use in many different ways.
So there's some questions here specifically about the future types of data we're going to collect, including dental, vision, and geospatial. Can you give us some thoughts on where that's going?
Wow, great question. The one of our sites we talk about having actually the dental records there too. As far as I know, some of those data are probably harder to get than others. I guess that's always a true statement. We are certainly thinking about geospatial kinds of data, both things that derive from surveys as well as EPA and other type measures that are of our physical and built environments. In terms of many of those other data sets, we have ideas around linking to all those things where many of those kinds of things we're definitely in our infancy in terms of what those plans are. This is a question I probably should have just thrown back to Sherry, to be honest, but I kept you off the hot seat.
Good, thank you. So the next question is sort of one about the publication protocol. So I think Kelsey will get a little bit more into this, but do folks have to apply to use the data and publish from all of us?
So there's she will go through that in detail. The shorter answer is you do have to go through an application process, but it's really unlike anyone that, that I've been involved with. If your institution's already signed a master agreement, which I don't know what the current count is, it's a big count, upwards of 150. If your institution's already signed that, then you could be on using the data today. And if you're really fast, you could start writing your paper tomorrow, I guess. And then you don't have to get approval for that paper. You don't have to get approval for any new project. It's a passport access model. Once you're approved as a researcher, you are approved to do anything that's within our bounds. You just have to describe it first, and then you get to work.
Okay, one final question. How are the EHR values normalized?
They are normalized according to the common data model. And so that is defining, if you're familiar with terms like LOINC, SNOMED, other vocabularies, that's trying to say that we're going to use the same vocabulary for them. We're going to have the same sets of units and things like this. I like saying EHR collection and curation is a journey, not a destination. This is a project, as COVID has taught us, just a case in point, that it's an evolving one. And, as we collect more data, there will be more efforts needed to do more normalization, as well as getting the old stuff better. So these are the kind of things that the team is keeping track of. And there are robust efforts both at all the different data providers, the team of, my data is a year old, but more than 180 people were on this EHR operations list a year ago, I remember. And I'm sure it's more now that are really working to curate, clean, and normalize these data to common standards to make them as usable as possible. So each version will get better. That's the commitment we'll make.
Okay, thank you so much, Josh. And we have several questions that weren't answered in the Q&A section. We're going to try to answer those directly in the Q&A session, so everyone's questions were answered. But at this time, I'd like to actually turn it over to Kelsey Mayo, who's from our Data Research Center, who's going to talk specifically about how the Research Hub works.
Thank you so much. Oh, go ahead, Raquel.
Oh, I was going to say, I will formally introduce Kelsey Mayo. Kelsey is a scientific product and portfolio manager here at Vanderbilt University Medical Center. Dr. Mayo is a multidisciplinary scientist with over 10 years of precision medicine research experience and a founding member of the All of Us Data and Research Center. Dr. Mayo recently led the successful development and launch of the Researcher Workbench, the All of Us Research Program's cloud-based analysis platform. She began her career as Beckman Scholar at the Georgia Institute of Technology and later earned her doctorate designing advanced therapeutics as an NSF graduate research fellow at Vanderbilt University. In her current role, Dr. Mayo is dedicated to partnering with the scientific community and finding creative solutions which accelerate biomedical research, innovation, and translation.
Thank you very much, Raquel. I appreciate the opportunity, as Raquel mentioned, to really come and join this community of researchers that we have on the call with us today to tell you a little bit more about some of the exciting things that we're doing in the program for researchers, and hopefully open this channel so that you join this ecosystem we're building, and you provide us feedback so that we, as Josh mentioned, can grow the program with you together. Next. So I always like to start by thanking the most important partners in this program, our participants. So thank you. Hopefully there are some participants even on the call today. Without them, none of the scientific advancements that we hope to enable through this program would be possible. We're also incredibly fortunate to work with a variety of community partners all across the country and really making an incredible network and a team that is recruiting, enrolling, and engaging with our participants to make this possible. Next slide. I'd also like to say thank you to our consortium partners. As Josh touched on earlier, this is a huge national effort. And in addition to our community partners and participants, all across the country, we have teams who are actively working to make this program possible. So thank you, everybody. Next slide. I'd also like to say thank you to my colleagues at the All of Us Data and Research Center, as well as at the NIH. So the All of Us Data and Research Center includes organizations such as Vanderbilt University Medical Center, who I represent today, the Broad Institute, Columbia University Medical Center, and Verily. And you can see a quick screenshot here of one of our recent face-to-face meetings, and even this isn't the entire team. So what I hope to impress upon the audience today is that this has been a tremendous collaborative effort, and I'm honored today to be presenting on behalf of a group of such talented people. Next slide. As Josh mentioned earlier, our mission in this program is to accelerate health research and medical breakthroughs for all of us. And we have three objectives that we really focus on to enable this mission. The first and foremost is nurturing relationships with our 1 million or more participant partners. The second objective, which will be the focus of my part of today's session, is to deliver the largest, richest biomedical data set, one which is easy, safe, and free to access. And then third, to catalyze a robust ecosystem that can use this and fully leverage this resource we are building to advance precision health. Next slide. So what I would like to orient you on, as Josh did a nice job of doing earlier on in the talk, is our research hub. So our research hub is how we are delivering on that second objective, the biomedical research data set. And you can see here in the top right, or excuse me, the top left-hand corner of the screen, a screenshot from our public website. And if you haven't already done so, I encourage you to visit researchallofus.org. You can see here our welcome message to all researchers, those who are in attendance today and future researchers to come. We call it our hub because this is really where we are going to connect you with the resources to access, analyze, and learn about All of Us participant data. So we're bringing you, the research community, into the cloud with us in order to leverage this amazing resource and really derive the maximum possible value that we can from the data that our participants have donated. And so you can see here, as you follow the chain from our Research Hub public website, you can apply for access and get what Josh mentioned earlier, our data passport. And once that access is granted, you are given access to the restricted portion of the hub, our All of Us Researcher Workbench, which we are thrilled launched this May in the beta phase. And it is within the workbench that you as a researcher will come in, learn about our data, access that data and the tools with which to analyze it, which we'll be going through today in this demo. Next slide. So briefly, what I hope to do today is walk you through an overview of our data and then go straight into a couple of live demonstrations, both of our public website, researchallofus.org, as well as our researcher workbench. Next slide. So let's start with the data overview. Next. As Josh mentioned, one of the most exciting things about this program is that our data are constantly growing and evolving. And of the data which are currently collected as part of the protocol for this program, electronic medical records, participant survey data, and physical measurements that were collected as part of the baseline assessment for this program are currently available in the research data set. So again, of all the data which are collected, these three data types are currently available for you to come in and perform research projects on. Next slide. And as Josh alluded to as well, we take the data that all of our participants and partners in this program provide to us as the Data and Research Center, and we carefully harmonize and curate this according to a common data model. So what we've chosen to do in this program is leverage the Odyssey Collaborative's OMOC common data model. And this common data model is particularly powerful because, as mentioned previously, it leverages standard vocabularies as well as a vast amount of metadata, so that once researchers get access to this resource, you really have a tremendous amount of flexibility in your approach to accessing and analyzing health information from our participants for your study. Next slide. And if you look at the research data set, which is currently available within the researcher workbench, you can see that, as Josh mentioned previously, we have well over 220,000 participants who have some survey data. This includes data from self-assessment of participant demographics, as well as personal medical history, family health history, et cetera. We also have physical measurement data, including anthropomorphic data such as height, weight, hip circumference, et cetera. And this is for the majority of participants in this data set, well over 180,000. And over 127,000 have also contributed some electronic health record data. At the right-hand side, I've tried to give you a sense for how many of these participants have contributed all three data types that are made available within this research data set. So we see that 126,000 plus participants in this first data set that we've made available have EHR data, physical measurement data, and survey data available for study. Next slide. Importantly, as we work to make this data available to you in the research community, we led a number of different demonstration projects, which will be delved into in detail in the second half of this program. But I'd like to touch just briefly on some of the early work that we did, which was led by Dr. Andrea Ramirez, where we were doing initial characterizations of this data. One of the more important findings from this project is that the participants included in this research data set are in fact diverse, with 77% meeting at least one of the underrepresented in biomedical research categories. You can see some of those listed below on the slide. Also incredibly important to this is that over 47% of the participants in this initial research data set are non-white or of Hispanic Latino ethnicity. Next slide. Another important thing that Dr. Ramirez and her team did was characterize this data set to ensure that we can reproduce known associations. And so here you can see a quick snapshot of the work this team did doing a phenome-wide association study looking at participants who are designated as ever smokers, either looking at data from their electronic medical records or data taken from their surveys, and looking at associations with increased risk effects. Here you see cancer of the lung, as one might expect, cancer of the respiratory system, as well as bladder. And so this gave us a lot of confidence as we moved to release this data set for research. As we can see, we accurately reproduce associations that were already well known in the literature. You can see the citation for this work below. We've published it as a preprint article on medRxiv. Next slide. And so I hope that you, following today's talk, are able to go and look at this paper, see the additional characterization and descriptive analyses that we performed. And I'd also like to call out the fact that once you get access as a researcher on this platform, we make the complete analysis available to you as one of our featured workspaces so that you can reproduce every aspect of the work that we're publishing. And this is really, I think, one of the powerful things about bringing you, the research community, into the cloud with us so that we can really improve the reproducibility of the science that we are doing. Next slide. And as Josh alluded to, we are so excited to be on the cusp of releasing a new version of this data set, which we will refresh data to get more recent data and more participants with the existing data types that I already covered. We are also adding data from 2 of the other sources that Josh touched on previously. We will be adding some data from our COVID-19 survey, the COPE survey. We are also going to be adding some pilot Fitbit data into this research data set very soon. Next slide. And to once again give you a sense for how the data will evolve in this next release, we expect to see approximately 40% increase in the volume of participants who have survey and physical measurement data. And because of the incredible work of the team who is actively curating and providing those electronic health records, we expect to see nearly a 60% increase in the number of electronic health records available for research. Also importantly, we have nearly 63,000 participants who have contributed data for this COVID-19 survey and 8,000 who have provided some Fitbit data. Next slide. So now that I've given you a quick sense of the data that are available for research, I'm about to transition into a live demonstration of both our public website as well as our researcher workbench. Before I do that, I'd like to do a few things to just orient you to what you're about to see. Next slide. So when I mention our public research or public website, this is our research hub, and you can see here a screenshot of our homepage. Next slide. While there's a wealth of information that I encourage everyone to check out and explore within the public website, in today's talk, I'm going to focus on one of our really exciting tools, our data browser, which hopefully you've had a chance to check out since the beginning of this meeting. This is really a fabulous resource if you want to explore the data that are currently available within the research data set. Next slide. I'm also going to touch briefly on our application process and point you to where to find more information on this public website. And for those of you who have registered to attend one of our breakout sessions later this afternoon, our team of experts is going to be walking through this in much more detail. Next slide. Assuming in this scenario that you've applied for access and we'll get access to this restricted portion of our website, our researcher workbench, I'll be walking you through a demo of the workbench itself. Next slide. Pointing out some of the helpful support resources that we integrate directly within the workbench and make available to you as a researcher. Next slide. And then quickly walk you through an example analysis in one of what we call our workspaces. Next slide. And so with that, I am going to transition again into this live demonstration. So bear with me one moment. I'm going to share my screen and confirm that you can see the new content. Okay. Can everybody see the screen that says, "Welcome all of us researchers"?
Yes.
Thank you. This is our public website. On our homepage, you can see that hopefully, as is coming clear through the course of today's talk, we truly do welcome you. We are open for business, so to speak. You know, we really would love for everybody attending the call today to apply for access and join the research community that we are building. On this public website, you'll also find helpful information such as snapshots of our progress overall, enrolling participants in the program and providing data. And what I'm about to go into is our data browser. But of course, before we do that, I'll quickly point out for those of you who, for instance, are survey researchers and are interested in some deep dive information into how our survey instruments were developed, you can also find more detailed information by exploring our data sources here. And so with that, let's go and check out our data browser tool. This is a live public tool, and it uses an aggregate public data set. So you'll notice here that all counts are aggregated, and this is done to protect participant privacy. And the goal of this tool is to allow you to explore and really understand what data might be available for research. So In this example, let's note that I am a principal investigator, and I'm particularly interested in the fact that all of us collects data from so many different sources. And since I know that all of us collects electronic health record data, as well as physical measurement data from an in-person assessment, I'm interested in doing a quick data quality comparison in some of the anthropomorphic data that is collected in this program. So let's look at height. If I wanted to just start by doing a generic search for height, I can search a keyword here. And then once I click enter, the tool will update and show me areas where there might be some potential matches to the thing that I'm interested in. So here I can see that there are program physical measurements which do capture height. And by digging a little bit deeper in this tool, I can see interesting distributions in height for different categories of sex assigned at birth, as well as age at which this measurement was taken. And if I'm curious, I can also explore other program collected physical measurements in this page, such again as weight, waist circumference, etc. If I go back here, and I look and see within the EHR data what might be of interest to my particular study, I'm going to explore further into our labs and measurement domain. So you can see here that for my specific search, I have a number of different participants who have contributed body height data as part of their electronic medical records. So I can explore this further if I would like to look at distributions such as distributions of values or sex assigned at birth, or since, as we've mentioned several times throughout today, this data is aggregated from a number of different sources. We can in fact look at the various sources of data that have provided this specific health information. And as a researcher, again, you can explore within our data browser a number of different things. But in this example, let's say that this gives me enough confidence that there is sufficient number of participants with data for my data quality study. And so as a researcher, my next step might be to apply for access. If I click here on our apply page, you'll be able to see a number of different pieces of information here which tell you about not only the application process, but also what it means to be in a beta phase. Because I'd like to emphasize that when we launched back in May of this year, we launched in a beta phase. And central to this concept is the fact that we are launching essentially small and opening our channels of communication with you as our research community, so that as we evolve, we can do so directly from your feedback. So we highly encourage you to provide us with feedback on both our data and our tools and our support services, so that we can continue to grow this resource with you. In this beta phase, an institutional agreement is required for access, as is an ERA Commons account. Again, we welcome your feedback. And we'll note that we are continually evolving both our tools as well as our data as this program evolves and as we collect more feedback. Our goal here is to be a true game changer for understanding health. And one of the things that we've done in the interest of shifting paradigms to be a game changer is really come up with a new application model, if you will, where we're assigning what we call passports here, where as a researcher, if you have an institutional agreement in place, you're able to complete a registration process, create your account, connect that account with an ERA Commons account, complete all of us responsible conduct of research training that was developed with partners in the program, and finally sign a data user code of conduct. And once this is complete, you'll be able to access our researcher workbench. And so with that, I would traditionally click apply now and go through each of these steps. But for the purposes of today's demonstration, what I will do instead is I'm going to click over to another tab here where you can imagine that I've been granted access as a researcher, and I now am within this restricted portion of the hub known as our All of Us Researcher Workbench. I would like to note that this is not our production website. This is a mirror of our production website that only contains synthetic data. So no real participant data will be shown during today's session. That being said, I'd like to sort of welcome everybody here to the All of Us Researcher Workbench. Because as a researcher in this example, I've already been very active within the workbench, you'll see a number of different features here. First and foremost are my workspaces. So within the workbench, your workspace is essentially your project. And as a researcher, as an approved researcher, when I want to study All of Us participant data, I have to carefully describe my intended study as part of creating a workspace. And I describe the scientific questions that I'm going to be addressing, my approach. And then when that is done, I've created a workspace which I can share with collaborators, and I can actively access and analyze data specific to that project. You can see on my screen that I've been active in 3 different projects recently. So I have one project where, as mentioned previously, I've been demonstrating anthropomorphic data quality, looking in detail into standing height. My colleague had also been doing a project on standing height and added me as a collaborator here. And so you can see that while I don't own this, I'm still an active collaborator on this project. And because I have another co-investigator that I work with who is an epidemiologist and endocrinologist, I've also been collaborating on a completely separate project looking at diabetes A1C values within the platform. So really this is what we hope is a beautiful way to organize your work and collaborate with your team to conduct these projects. If I had just been a brand new user, I'd never been introduced to a collaborator in this space, I'm coming in fresh, you wouldn't see these cards because they would not have been created. Instead, we'd have pointed you to a number of different helpful resources. And let me show you a few of those below. So one of the things that our team has done is put together a number of different help materials, including tutorial videos, giving general introductions to the researcher workbench, as well as the tools that researchers have access to in order to analyze and subset data for analysis. All of those tools are made available within the workbench. And I'd also like to quickly orient you to other support and help materials which we provide to our research community. So if I click here at the top left-hand corner, we call this the hamburger menu, if you will, you can see that while I do have access to my specific workspaces, as I described earlier on in this session, you also have access to our featured workspace library. This is a really important, helpful resource because we are curating a set of example analyses, tutorials, and fully implemented electronic phenotypes that we are making available to all researchers within the workbench to better help you do your work. We also have an integrated support hub here, so I'll click this just to give you a sense of the type of resources which we make available within our support hub. So you can search an entire knowledge base that our team has carefully curated for you. You can also access helpful information under Getting Started that give you details about our All of Us data set, our privacy methodology, curation process, as well as tutorials on how to use specific tools. So this is really a powerful resource for any researchers who are getting started working within the platform. Let me go back to the researcher workbench here and also note that if at any time throughout your project, you've searched the knowledge base, but you haven't found the answer to your specific question, you can always contact us directly through this help desk function here. And we have an incredible team of staff who are knowledgeable in our data and tools and program to help answer your questions. So now that we've oriented just generally to the researcher workbench, I'd like to transition into the portion of our project where we're going through an example of analysis. So let's look at a workspace that my colleagues and I have been working within. When I go into a specific workspace, we organize things into tools for subsetting data, tools for analyzing data, and of course, a tool that lets you look at your project generally, the purpose of the project that you've previously described, collaborators, etc. Let me spend just a moment here to note that this is an incredibly important part of the research experience. As a researcher, we're asking you to tell us what you are doing with this data, and we are promising to make some of this information available publicly as part of our transparency with our funding agencies, as well as with our participant partners. And so you can see here that this specific project, as described a few minutes ago, the goal of this project is really, I want to compare the distribution of heights collected from these program baseline physical measurements to that data which was collected from electronic health records. I also want to point out that I have a number of different collaborators. I have a co-investigator. I also have a data scientist here who's collaborating with me, in addition to a few graduate students on this project. And I can see other information. Importantly here, you can see that I have spent $4 of my free credits. And this gives me an opportunity to highlight one of the important things about the beta phase. Right now, we are offering free credits for anybody who chooses to participate as a beta researcher within the workbench. Access, as mentioned previously, is free. However, since this is a cloud environment and you will be doing your analysis in the cloud on some of the virtual machines that we make available to you, compute does cost money. It's very inexpensive relative to a lot of traditional models, but to offset that cost, we are making free credits available. So let's go and look back at some of the tools that we have for subsetting data. So at this time, I hope that you've gotten a sense for how large even this initial release of data is, and will only be growing and getting bigger all the time. And we recognize that it's very important for researchers to be able to find from within the larger cohort a more specific group of participants, a cohort of participants that have some shared criteria that's relevant for your analysis. In order to make this easier for you, we have created a tool we call our Cohort Builder tool that allows you to select and subset participants. So in this example, let's keep going with the idea that I would like to select participants who have some physical measurement data and specifically height data. So I'm building up my criteria here and specifying that I would like participants who have some height information. And because this is a measurement, I'm given the opportunity, if it's relevant to my project, to specify certain ranges of height that are important for my cohort selection. In this case, I'm going to keep it very simple and say that any value is sufficient for my analysis. And I'm going to add this criteria, save it. And then at the right-hand side here, you're going to see my cohort of synthetic participants update and give me additional information about overall descriptive statistics or demographics in this case. And so here you can see the distribution of gender identities of participants within the cohort, as well as breakdown of things such as age and race. Within this synthetic data set, there are nearly 200,000 participants who have some physical measurement data from the program baseline assessment, but I'd also like to ensure that my cohort has some data from the electronic medical records. So now I'm going to build up this Boolean logic string here and say I would like to select participants who have height measurements from their EHR data. And in order to do this, I can look at, for instance, body height as measured by a variety of different concepts here. This is where you start to see the power of our common data model. So we can see that there are a number of different ways that body height is captured within the electronic medical record. And in this example, if you look at this SNOMED term, the SNOMED vocabulary, we know that this is very hierarchical vocabulary. And so it might be helpful here to switch the view from a simple list to look at the hierarchy of information and the context of this specific health concept within this vocabulary. What this, we hope, does for our researcher community is allows you more fine-tuned control of how you're building and selecting participants for your analysis. And so if I go back to the list view here, I notice that this particular concept does not have very many synthetic participants. So let me just explore this concept here. I'm again going to say that any value for this particular measurement is sufficient for my analysis. But because I know this comes from my electronic medical record, I also have some neat ability to modify my concept and say, you know what, I don't want just one mention of this in my electronic medical record. I'd like 2 or more mentions of this. And I'd like to ensure that this was taken as part of an inpatient visit at the hospital. And so again, this is just more for demonstrative purposes than anything. This tool, however, does allow you a lot more powerful capabilities to fine tune the controls for your criteria selection when building a cohort. So let's save this new criteria. And now what the sequence says is, I want to select participants who have some physical measurement data from program assessment, as well as have some electronic health record measurement of height, 2 or more occurrences of that in their medical record, and the height had to have been taken during an inpatient visit. Because this is ANDed together, you'll see that the total count of participants who meet my criteria is about 9,000, and you can see that I've updated the distribution here according to the gender identity, age, and race of those 9,000 participants. So let me quickly create this cohort. I can describe my cohort in as many words as make sense for my analysis. And then once I save this, I'm given a number of different prompts for my next step. So let's imagine that, unlike this current example, I were doing a case control study. Rather than proceed to review the participants that are in the cohort I just created, it might be logical for me to go back and create a control set. So I do have the ability to create another cohort. Because I will not do that in this example, I'm going to go straight and create what we call a review set. We built this capability for those that are interested in doing detailed electronic phenotyping within the All of Us data set, particularly using something which is a complex data type such as the electronic health record. So to facilitate this research use case, we allow you to select a random sample of the participants, in this case from the 9,000 that met my criteria that we discussed previously. And I'm going to get participant level information on these participants in my cohort. So you can see here a listing, it'll be of 100 participants. I get some summary information here of their demographics, and I also get the ability to dive more deeply into the data which we have available on these participants. Because you see the date of birth column here, I'd like to clarify, as Josh mentioned previously, our registered tier data set is a de-identified data set, and dates within this data set are shifted. They are date shifted. However, what we have done is date shift consistently within an individual participant record to protect the ability to study the temporality between events in an individual's record. However, across the entire cohort, all dates are shifted randomly. And so we recognize that this does prohibit certain research on things that have seasonality to them, which is why we are so excited about eventually being able to provide the next tier of data that Josh mentioned, that controlled tier, which also has things such as our genomic data. So that being said, let's look at one of these records here. I'll just select the first one for the purposes of this example. If I've been in the process of doing detailed electronic phenotyping, I'll be able to go through this data more specifically and see if this participant really does seem to meet my phenotype of interest. And if not, I can refine different things. rather than wait for this to load, let me go back to the review set and show one more important feature. I'd like to highlight the fact that this tool allows you to see a very clear description of the cohort that I just created at any time. So you can always come back here and see key descriptive statistics and even get a glimpse of top things in the record. So top conditions, top medications and procedures, et cetera. And this basically saves a snapshot of the work that I've done, which is particularly important. Let's say if this was done by my graduate student, and as a principal investigator, I'm reviewing their methodology. I'm looking just at the highlights of the work that they've done to see if we want to proceed and create a data set using this cohort of participants. So let's assume that I've done that. You know, my graduate student came in, they created that cohort, and as I reviewed that cohort, it met the criteria for my research study. Now I'm interested in creating a data set of information that I can work with my team to analyze and really understand the data quality between our height information from these two different sources. So if we look within the data set builder tool, I have now got the ability to combine the cohort that I have just created with other health information that might be relevant to my study in order to generate a data set for analysis. So we do provide some prepackaged cohorts here, such as the All of Us, All Participants cohort. But once this is finished loading, you'll be able to see as well the custom cohorts that I have created, as well as any custom groups of health information that my team has created for study. So let's walk through the example with one of these prepackaged cohorts here. If I want to select all participants, and just an example here, and their demographic information, I'm going to see that I can also really carefully select what columns of information are going to be within the data set that I will export for analysis. I can also change my mind and say that I would like to use not the all participant cohort, but rather one of the specific cohorts that my team has created. So here's the one that my graduate student created and I reviewed and looks great for this specific analysis, my height cohort. I can also access, their demographic information here, or if I want to get specific information other than demographics, select from a custom created set of health information here we call a concept set. The process for creating a concept set now looks very similar to the process for selecting criteria, so I'm not going to go into that during today's demonstration. Instead, let's assume that my team created this and I'd like to use it in analysis. We can preview the information that we're about to take into an analysis tool in just a moment. And you can see that because I selected demographic information and some height information, the tool will break this into two different data frames for study. One, which is comes from our person table, has all of this demographic information. One, which contains all of the height information for study. If this data looks good and I don't want to further customize the tables for export, I can save and analyze this new data set. And you'll see here that after naming my data set, I'm asked whether or not I want to create a new notebook in which to place this data set or add it to an existing notebook. So what I'm about to go through in the next portion of the demonstration is a much more detailed overview of the current analysis tool that we have available. We call these Jupyter notebooks, hence the name notebook. And we currently have support for programming in either R or Python. And so if your team is most comfortable in programming with Python or R, you could specify which of those two languages and then create a notebook in which to do your analysis. So I just wanted to show you what it looks like if I'm creating a fresh new notebook. What you see on the screen here is a query. It is code that the tool created for me as a researcher. So when I was doing all that pointing and clicking, behind the scenes, our tool was creating a query that would allow me to pull a data frame for analysis. So that's one of the powerful things about those point and click tools is not only do we create the query for you, but we let you see exactly what the query was to try and provide transparency into the way that we are doing, the way that we are extracting data from our data set. Rather than go into this new notebook, let me just quickly touch on one that, again, a colleague of mine had been working on in this workspace to give you a sense of beyond code, what the incredible capabilities are of Jupyter Notebooks to support reproducible science. And so here in this quick preview, you'll get a sense for this, whereas previously you saw some lines of code, here you can see human readable language that clearly describes the work that my team has been doing in this analysis notebook. And I have the ability here as a PI, because I actually don't want to edit any of their work. I just want to play around in the notebook and not save any of my changes. So I'm going to open this Jupyter Notebook in a playground mode here. And you can see in those steps as the screen was loading that what has been done is the researcher workbench is spinning up an analysis environment for me as a researcher. And as it spun up this analysis environment, essentially partnered this notebook you see on the screen with a virtual machine that allows me to conduct my analysis. And so when I look at what my team has been doing, they've been describing their methodology very carefully. I can see, for instance, how they used code from that data set builder in this notebook. And so you can see the output data set, but let me show just quickly, here's the code. So as I showed you in that previous screen, all of this was generated for you by the tools themselves. And then once that data frame is available within my notebook, I can go forward and I can do additional data cleaning steps and exploratory data analysis. And here on the screen, you can see a demonstration of another thing that our team has provided for you, which we call code snippet. So as part of our exploratory data analysis, we've created a library. You can see the video here, but also within this top menu of groups of code that you can basically drag and drop into your notebook. And with changing one or two small lines of code, run some early exploratory analysis, some graphs such as height by gender, race, etc. So let me just quickly scroll down and you'll see that I can not only describe what I'm doing in human readable language, I can also write my code, execute my code, and see the outputs from my code all in a single notebook. And as a researcher who sort of grew up in the world of MATLAB and other programming languages where I had my entire project scattered across different things, you know, Word documents, Excel, MATLAB, RStudio, et cetera. I think, you know, RStudio being another example here of how the entire field is evolving to try and bring these things together in one spot to support reproducibility and fair science. And so we're thrilled, again, to be able to offer this as the initial analysis tool because of its flexibility and its ability to support multiple programming languages, et cetera. But I also will note that part of us evolving this platform is that we're actively looking to partner with the scientific community and see what other analysis tools we can make available within the researcher workbench to support the different scientific use cases that we would like to enable. So as sort of a punchline here, and then I'll wrap up the demonstration part of my talk, you can see that when I ran this analysis with the two different sources of height information coming from both survey data and electronic health record data, we do see what we would have expected, and that's some pretty similar distributions. Now, while this is synthetic data, our team did in fact run this entire thing on the real data set as part of some of our early quality work. And though I've been describing this notebook as if it were something that my team collaborated on a research project, it's actually one of the featured workspaces that we make available. So I pulled it into this notebook for the purposes of demonstration, but this is the level of detail that our team has gone through to try and provide you with resources that teach you how to really work with the different data types that we are making available. So in case you have access to the Researcher Workbench, go ahead and check out our Working with Physical Measurements Data featured workspace, and you'll be able to run this entire notebook yourself. And so with that, I'm going to stop my screen share. Raquel, would you mind taking back over and sharing the slides? Thank you. Okay. So again, this was just the live demonstration that we just went through. Next slide. And today, during my portion of the talk, we talked about the data which are available currently and give you a quick sneak peek of what's coming very soon. Then we showed you our public website, as well as our Researcher Workbench itself. And that was just a mirror, again, not our true production environment. But sort of the key takeaway here is that we hope you're interested in joining us as one of our beta researchers in our growing community. And if you would like to do so, we would like you to visit our public website and click that apply button and go ahead and start that process. And hopefully many of you are also registered to do one of our breakout sessions towards the end of the afternoon, where we'll be going through that process in more detail. Next slide. Before we break for first questions and then give you a true break to get up and stretch, I'd also like to highlight briefly, we've got some incredible presentations coming up from partners in the consortium who had led some amazing demonstration work and have really seeded the incredible community that we hope to build. And so we have a presentation, a keynote address coming up from Dr. Lucila Ono Machado, as well as detailed overviews of projects by Dr. Jason Carnes, as well as Dr. Mitch Lunn. And then following that formal session, we will be breaking out into our different onboarding walkthrough sessions. So we hope you stay with us. It's been really an honor talking with you today, and I'm excited to see what comes up in the chat for the next few minutes. So please send us your questions. Thank you.
I will unmute. The first question that we have in the chat is, can one enter ICD-10 codes in the data section?
Yes, I should have demonstrated that, but you don't just have to do a keyword search. You can search for a source code, like you mentioned here, a ICD-9 or ICD-10 code. Or if you wanted to search by a standard code, a particular LOINC code or SNOMED code, we do allow that flexibility, yes.
And are GPUs available on the workbench?
Yes, every researcher is assigned a virtual machine. I think it has 26 gigabytes of RAM by default and 4 cores. And that is something which, should your project require a more powerful VM, you can apply for one in collaboration with our help desk. But we found that that has been sufficient for the current data that we have available within the Researcher Workbench.
Okay. And then another question we have in the chat is, did I read that slide correctly that 8,000 people out of 300,000 are contributing Fitbit data? Or is that just a subsample to be released in the pilot and the percentage is higher?
So in this upcoming release of data, we will be making available approximately, I guess it's a little over 8,000 participants who have Fitbit data. This is essentially part of a pilot effort. So this is the earliest release of that data in the program. And that is out of the 315,000 or so who have contributed data in that upcoming data set.
Okay, next question we have is, do you have technical support for installation of non-standard R packages that might require additional configuration?
Yes. And we can get into this a little bit more detail offline as well. But essentially, we have the ability to import libraries of packages, software packages, both in R and Python that are necessary for analysis. And our team is there to help you if you have any questions or running into any issues doing that. And we've worked with researchers before to make sure that we can meet their research use cases.
Next question we have is, could several folks each register programmers, grad students, investigators, and coordinate to sort the pool of their $300 free credits so that they can apply them to the same project?
That's a great question. Our current model with free credits is that whoever created the workspace and is the original owner of that workspace is who the free credits are deducted from. But there are strategies that you could pursue to help share that free credit and spending. One of the things that I didn't mention in the formal part of the talk that is important is that the last time our team ran the analysis about a month ago, in total, all approximately 300 or so registered researchers that we have in the workbench have cumulatively spent less than $1,000. And I think 90% of our researchers have spent about $15 or less cumulatively for all their projects. So it's part of the way that we really hope to lower the barriers to doing this type of big data research by putting it in the cloud and lowering the cost of doing this. So right now, we really haven't seen any issues with free credit spending.
Next question we have is, can I use a Docker R or bioconductor image?
Let me take that one back to the team to get you an affirmative answer there. I think the answer might be no currently, but our team can follow up after the program.
Okay. Can you please clarify if all data analyses have to be conducted on the servers, or can a curated data set be downloaded for analysis on a local machine using statistical programs beyond R and Python that seem to be the only options available now on all of Us Researcher Workbench space?
Yes, so the answer is you cannot download participant level data locally. All research done on participant level data must be done per our data user code of conduct and data use agreements within the researcher workbench. And we do recognize that that presents challenges for researchers who are most familiar with working with SAS or Stata or SPSS, some of these other tools. And so we understand that that does make certain aspects more challenging, but really the benefits of doing research in the cloud outweigh the ability to download this locally. Number one, at some point, this data set is going to be too big, essentially, for a lot of researchers to work with locally and very expensive to store many copies of. But also, this gives us greater control over audit and security and privacy, which are fundamental to this program and protecting our participants. And so it's for that reason that we ask people to do we require people to do their research within the researcher workbench environment and only permit download of data, which is aggregated or figures example with aggregate statistics and data that would be used for a publication.
And then we have an answer from Shimon Rura in our chat box about the Docker images. So no custom Docker images for the foreseeable future. And we don't yet have GPU capability in the workbench, but it may be added later.
Perfect. Thank you, Shimon.
Next question we have is from Melissa. We've had a very hard time merging data across surveys wherein participants' data across surveys are matched with one row per participant.
So one of the things that if you haven't already done so, I'd love for you to check out the featured workspace that we created on working with all of us participant data, specifically from surveys. And I think that we might have covered a few of those use cases there. If not, please reach out through the community forum that we've built. Our team also monitors that community forum, and we can help work through some of these challenges with you, as can others in the Researcher Workbench community.
Next question we have is, do your survey data include promise items, For example, the Promise 29, and are these measured only at one point in time or portions of the survey repeated regularly over time?
Okay. I will defer parts of this answer to, I know we have experts behind the scenes here who might be able to answer this in the QA box. What I will encourage you to do is visit that Survey Explorer within our public website. So for each of the survey instruments, which are available for research now, there's a detailed walkthrough of where specific questions were sourced from, any modifications that the program might have made in order to develop that survey instrument. And then finally, I would like to note that the only longitudinal survey which we've released to collect repeated measures to date is our COVID survey, the COPE survey. So that's going to be the first repeated measure in terms of our survey data. However, we will be looking at making other surveys, which we currently collect, repeated measures and to do some more longitudinal studies with participants over time.
Okay, next question we have is, can we enter a DEAN or a S&P RS number?
Okay, excellent. Right now within the Researcher Workbench, we do not have any All of Us program genomic data. And so we do not provide support yet for searching through SNPs and variants within some of our point and click tools. However, we are actively working to develop those and release those, as Josh mentioned, hopefully about a year from now, for scientists who are part of the Researcher Workbench community.
Okay, next question is, I wasn't clear about the dental EHR data. Are you working on that now? And if so, could you tell me who your partners are?
I might push that one to either Josh or Sherry, if either of you are on the line to answer.
I'm still here. We haven't made any dental EHRs at this point, are not something that we have much thought or progress into. I will say that there are a few of the of our EHR providers that actually have dental information as part of their EHRs. So it is possible that could be an element for which some of that could be imported. But as far as a national, kind of a more broad national strategy at this point, we don't have a lot of knowledge in that space. But if you have suggestions on how those data could be retrieved or shared, it certainly would be welcome to receive those ideas.
Okay, next question we have is, we can't download data, but you can upload data, but can you upload data? For example, genotype data for a disease with the idea of using the All of Us data set as controls. And by the way, fantastic progress in platform.
Thank you so much for the compliment. We're excited to hear that type of feedback. As far as can you upload data, the short answer is yes, but we have policies and restrictions on what is allowed. And so rather than reiterate that here in detail, I'll point you to I believe we have a policies section within our public website where you can read some of our policies word for word. But essentially, you as a researcher are taking responsibility for the data that you're uploading, that you're meeting the necessary data use restrictions. And so using an example, if I had a local map that I wanted to use for doing a phenome-wide association study, I could certainly upload that information into my workspace where I'd have a Google bucket and I can pull that into a notebook in order to conduct my phenome-wide association study. So there are certain types of data which you can bring in to support your analysis. Another example would be if you're doing a comparison of our survey data with survey results from NHANES, you could pull in some aggregate statistics from NHANES, for instance, to support like a side-by-side comparison of aggregate information from all of us.
Okay, our next question is from Patricia. How do we apply to conduct a study that involves recontacting all of us participants?
Excellent question, Patricia. I'll give a brief answer and then open it up if either Josh or Sherry would like to comment. The program is designed to enable recontact of participants in the future for other studies. However, we are not there yet. That is a goal of the program and is on our roadmap, but we do not provide support for that yet. And Josh or Sherry, please feel free to chime in.
Kelsey, that's a great answer. I'll just add that we have approved through the Council of Councils concept clearance for our what will probably be our first ancillary study that does have recontact as a part, and that's a study on precision nutrition. It's called Nutrition for Precision Health, and you'll be hearing more information about that, but it has a series of where sub-cohorts will be pulled out, as well as people potentially enrolling specifically for the Nutrition and Precision Health study into the AOU platform. We'll have a series of studies all the way from just detailed dietary reporting to intensive metabolic studies, and certainly would include other kinds of omics and companion studies as well. So we're using this as a driver for thinking about ancillary studies more broadly, and certainly is something that we have on the roadmap.
Okay, great. And then I think we're close to being done. How does All of Us connect with Anvil or Terra? Anvil has provides a cost estimate of jobs, which is useful.
That's an excellent question. So we at the Data and Research Center are, as I mentioned previously, a collaborative team that includes partners from the Broad Institute and Verily, who are also incredibly active in these other projects such as Anvil, Biodata Catalyst, and other big data biomedical research projects, which are supported through a platform called Terra. All of Us, like some of those other studies, is essentially powered by Terra. So behind the scenes, we also use the Terra platform to enable research in the cloud. And we have been working with the teams at Terra to take certain functionality, which they built for other programs, and actually make that available to researchers and All of Us as well. It's one of the beautiful things about being a part of this biosphere of big data projects. And so while we do not yet have support for that cost estimate, we also think that's an exciting feature, and can't wait to be able to enable that in All of Us. And fortunately, that path is eased by the fact that we leverage the same platform behind the scenes.
Okay, one more question, actually two. Once genomics data is coming in, will there be something like a genome browser component in Workbench, similar to IUCSD genome browser?
The short answer is yes. And in fact, we're still taking sort of feedback and recommendations from the community on your favorite tools and things that you find most helpful to support your genomics needs. And so either through the chat mechanisms here or through our support email addresses, we welcome that feedback. So please feel free to send it our way.
And I think this is our last question. Will old data sets be archived? For example, if we use this data, will we be able to say we used All of Us version or release number XXX, et cetera, so that people know which set was used?
Yes, and we hope to provide support so that if you chose to do so, you could actually publish your work internally to our featured workspace library. And so not only could you reference the data set that you used, but in fact, you could make your entire analysis available to the community of researchers, which we think is an exciting way of driving forward science together.
Okay, so we will continue to answer questions in the Q&A chat. Unfortunately, we are running out of time. So we will be taking a short 10-minute break. If everyone can be back at 12:47, that would be great. Other than that, I don't know, Kelsey, if you have any other comments or anything else you wanted to share. Okay.
Thank you, everybody.
Great.
And exciting talks coming up, so stay tuned.
So I'm waiting for your go ahead.
Sorry, I just realized I started introducing you while being on mute. Sorry about that. We will start all over again. Today we have Dr. Lucila Ano Machado, who will be presenting a perspective from an informatics researcher. Dr. Lucila Ano Machado received her medical degree from the University of Sao Paulo and her doctoral degree in medical information sciences and computer science from Stanford. She is Associate Dean for Informatics and Technology and the founding chair of the University of California, San Diego Health Department of Biomedical Informatics at UCSD, where she leads a group of faculty with diverse backgrounds in medicine, nursing, informatics, and computer science. Also, she is the PI for the California Precision Medicine Consortium for the National Institutes of Health Us Research Program. Prior to her current position, she was faculty at Brigham and Women's Hospital, Harvard Medical School, and affiliated with the MIT Division of Health Sciences and Technology. Dr. Ano Machado is an elected member of the American College of Medical Informatics, the American Institute of Medical and Biological Engineering, the American Society for Clinical Investigation, and the National Academy of Medicine. She served as editor-in-chief for the Journal of the American Medical Informatics Association from 2011 to 2018. She directs the Patient-Centered Scalable National Network for Effectiveness Research, a large clinical data research network covering more than 30 million patients and 12 healthcare systems, and was once the founders of UC Research Exchange, a clinical data research network that connected the data warehouses of the 5 University of California medical centers. She was the director of the NIH-funded National Center for Biomedical Computing, IDASH, integrating data for analysis, anonymization and sharing, based at UCSD with collaborators in multiple institutions as well as other NIH-funded consortia and research projects. Her research focuses on privacy preserving distributed analytics for healthcare and biomedical sciences. She has received numerous awards for innovations in biomedical informatics. We're so glad to have you here, Dr. Lucilla, and I will let you get started.
Oh, thank you, Raquel, for the introduction, and thank you all for attending. It's very exciting to see this many participants, and it's also very exciting that this resource is becoming available to so many people. So my role here will be to describe our experience and also the process by which this demonstration projects got started. So in the next slide, you will see the charter for the demonstration project task force, which was to demonstrate the quality, utility, and diversity of all of Us research program data and tools via a partnership with the awardees. So we wanted fully executed research projects demonstrating the utility and validity of the data, and potentially to publish, but not novel discovery work, because now that the resources available to everyone can do discovery work. So the next slide showed the composition of the task force, and I want to thank them all, and particularly the chair, Andrea Ramirez, who kept us all in sync about selecting projects and monitoring progress over time. The demonstration projects subcommittee or task force is a branches out of the science committee of the All of Us program, and there are many other task forces as well. So the goal here was to replicate known disease and risk factor associations or validate predictive models that were already published. Implementation of phenotype algorithms and cohort characterization were important so that we could test the resource before it became widely available. So the next slide shows the phase 2 demonstration projects, the ones that I'm referring to, started with 44 submissions, and 33 of them were okay to proceed, and many are now in the phase of generating the papers. So the next slide showed again that the demonstration projects were conceived to test the platform, to provide constructive feedback to the program so that the resource could be enhanced, and investigate reproducibility of previous studies or statistics. So there were internal meetings to organize the projects, and also to interact with the Data Research Center on specifics of debugging and other aspects. There were general meetings where we all could share experiences, and now the publications are under review. The next slide shows the good-looking people who were involved in the launching of the projects. They were from several different sites that I'll show next. And it was very exciting to have this start of a big era of sharing data and making discoveries collectively. The next slide shows the anticipated timeline with, of course, some delays given the COVID crisis and so on. So we are at the phase that the teams are starting to publish phase 2 manuscripts. And the next phase would be control tiered, as was shown before. And this, so watch out for those, because it will be as exciting as the launching of this research workbench. The next slide shows that many groups were involved. And the areas, the health areas were many, with a predominance in cardiovascular disease, cancer, and diabetes, but also representing some other ones that are shown here, and the consortium awardees that were involved in this projects. And you will see two particular and more detailed presentations later today. The next slide shows some examples of proposals that were not accepted. And I just put this here because these were my own And you might say, well, you knew what was going on. You're still proposing things that were not doable. And the answer is that data may not be available at a time, but they will be available later on. So we ask people to be patient and to understand that it's an evolution of data that will happen. In our cases here, the proposal could be constructed as beyond the reproducibility or replication of results, and therefore it was too premature to be proposed at the time. So the next slide shows the demo, what you do to know if all of us has the data that you need. So as Kelsey shown, there is a data browser, and you can check that for the existence of the variable that you're looking for and count the counts of participants that have values. You can also check the Workbench's data dictionary if you want more specifics. And then the cohort definitional library to see if someone did the hard work of defining that particular cohort, which would be important if you are comparing your results from someone else's, for example. And then to check the notebooks with code that you can reuse. That will also accelerate quite a bit your analysis. So that's the spirit of having this common data commons that everyone can have access to. So the next slide, I will start the quick overview of demo projects. I will not present the topics of them all, but just to give you an idea of what went on in this phase 2. So the next slide shows one example that I picked of the Asian Health Coalition project, in which they wanted to characterize the prevalence of type 2 diabetes in Asian American cohorts of the All of Us Research program. They describe the background, why this is important, and why this population is different in terms of type 2 diabetes prevalence. The reason I wanted to show this one in particular, if you skip to the next slide, is that the cohort selection was very well defined. And again, you can reproduce that for your own research if you're interested. And then the next slide, also, the variables that were selected were, in this case, from the physical measurements, the PPI, which means the surveys, the basic survey and overall health surveys. And the reason this is important is then you know where the sources are from, and then Kelsey and others have shown already how many data points you have in there. So it's important to verify all these things before you decide which variables you want to work with. So the next slide shows also that this Asian Health Coalition had another project on demographic characteristics of the Asian American cohort, and also conducted that study. And I'll go quickly over the next ones just to go over the topics that they cover. So the next one from the Illinois Precision Medicine project was focused on hypertension and a comparison with the NHANES data set. And this was one of the projects. The next one that the group had also in the next slide shows a different topic of cohort characterization of mammography rates in All of Us programs. So you can see how diverse the projects are even within a given consortium like the Illinois one. The next one was the New England Precision Medicine Consortium, in which they studied, again, the prevalence of hypertension using EHR definition from the eMERGE Consortium among underrepresented in biomedical research groups, also with comparisons to NHANES. In treatment pathways using sequencing analysis were examined as well. The New England Consortium also had two other projects. The next one relates, in the next slide, relates to obesity in overweight. also among underrepresented groups. So you can see there is a lot that could be done even when we were restricted of not doing new discoveries, just to know what was in the All of Us database and characterize that. A third project from New England showed, examined uterine fibroids in the All of Us population. So this is to give an example. The New York City Precision Medicine had other projects, and again, focused on blood pressure and stroke in diverse populations. So taking advantage of All of Us research program having such diverse participants. The Southeast Enrollment Center focused on atrial fibrillation and examining that, again, using EHR in other aspects of the data that we have. Next, the Trans-American Consortium for Healthcare Systems Research Network, or TAG, studied opioid use in All of Us research program. Another set of projects was proposed by the Data Research Center, shown next, in which one of them referred to exploration of pediatric data from the All of Us program. And you may ask, well, the program does not yet have pediatric participants. How is that possible? It's because when patients enroll, the EHRs from childhood may come along. And the DRC was investigating how many of those there were, what data types are there, and could this be used for pediatric research? So that's another project. The other one from the DRC, or the other two from the DRC, were on more rare diseases and phenotype risk scores for Mendelian diseases such as cystic fibrosis, hemochromatosis, and sickle cell anemia. And yet another one on comparing family health history gathered in survey and EHRs for this registered here data. So you can see the variety of projects that were involved. So in the next slide, I will give the microphone to Dr. Sally Baxter, who will run over briefly on the California Precision Medicine project. So Sally, please get started.
Okay, great. Thanks, Lucilla. Yeah, I'll just give a few details about our project. I know we're rather time limited, so I won't go into all the specifics. If you're interested, you can always chat me or email me. But essentially, our thought here was to do some predictive modeling for glaucoma, which is the world's leading cause of irreversible blindness. It involves progressive damage to the optic nerve. And on the right-hand side of the slide, you can see visual field testing that we typically do in ophthalmology to monitor the progression of this disease. So on the next slide, we'll go over the study question, which was really to examine how systemic conditions like blood pressure and heart disease and other comorbidities, how systemic comorbidities and medications might affect glaucoma progression. And the pictures in the middle show some, pictures of different types of glaucoma surgeries. And so we used the need for surgery as a proxy for glaucoma progression. And hopefully all of you have eaten lunch already before seeing these pictures. And we'll go to the next slide, please. So we had a prior published predictive model that demonstrated that systemic data in electronic health records had some predictive value in identifying patients with glaucoma at risk of progression to surgical intervention, even without eye-specific endpoints. And some of the predictors that came out as significant in that model included blood pressure-related metrics and certain medication classes. We often don't incorporate these data in our routine clinical decision-making as ophthalmologists. And so the idea for this model was to see whether, we could use predictive models to understand the role of those factors and achieve improved precision management of these patients. So next slide. So this is just a table going over some of those top predictors. Next slide. So the demonstration project was really aimed at validating the prior model using all of us participants' EHR data. So we defined the cohort with the same criteria as the original cohort, and these were based on ICD diagnosis codes. We used Workbench to build data sets based on queries for predictors in the model. We did have to develop some custom SQL queries outside of just the Workbench interface and put those directly in the notebook for things like visits and medications, since we looked at medication classes rather than individual medication IDs. And finally, we cleaned the data and put it in the same format that we had used for the original model. We used the all of us data set as a test data set for essentially external validation of the initial model. The next slide. This just shows some of the workflow that we had. At the time, there were around 240,000 participants in the All of Us research program, and we narrowed this down to the patients or participants with glaucoma with the same ICD codes that we had used in the original paper. And again, we had done external validation of the single center model that we had published previously. And we also were curious to see whether if we used the All of Us data set as for training new models, how the performance metrics would be impacted. Next slide. And so one of the things we're interested in was looking at the diversity of the cohorts. And so if you look at the single center cohort, that's the one from our single institution that we previously published, a slightly older group than the All of Us cohort. And just stepping back a little bit, the All of Us cohort was much larger in size than our single center cohort, which makes sense since we're drawing from national scale across multiple enrollment sites. There was better representation of females in the All of Us cohort. And then you can see as far as the racial diversity goes, we had much better representation of Black or African American participants. And so, you know, that was another real strength of the All of Us cohort. The percentage of patients who underwent surgery was less, which was not surprising since our single center cohort was based off of a tertiary referral center with a high volume of glaucoma surgery versus the All of Us cohort is a more general group of enrollment sites. Next slide. So looking at the models that we trained with the All of Us data, we were able to achieve really excellent predictive performance, especially for neural networks and random forest. The logistic regression was more modest predictive performance, but still superior to what we were able to achieve with the single center model that we had previously published, which was sort of in the 0.67 to 0.7 range as far as the AUC goes. And when we use the model that we had developed at the single center and applied it to All of Us data, the AUC wasn't great. It was in the 0.5 to 0.6 range, which showed that our single center model was not very generalizable and did not perform as well when applied to a new test set that was external to the original data. And here we had done cross-validation on the All of Us data and had really achieved much better performance. And so I think this really was a demonstration that having a more diverse cohort that comes from multiple centers could result in models that perform better than those derived from single centers alone. And the next slide. So just in the final minute here, I'll talk about what we found as sort of strengths and maybe some of the limitations of this data, particularly for ophthalmic research. Really, there was, it's a nationwide scope. It's a very large cohort. The emphasis on underrepresented populations is great for looking at questions where we may not have had great representation in prior clinical research studies. There's a huge range of data types. And in our paper, we were mostly focused on electronic health record data, but future work, we plan to look at social determinants and survey data and so forth. For limitations, there are still some constraints to the All of Us data. For ophthalmology, as an example, ophthalmic observations in the data warehouse were pretty sparse. And like I mentioned in the first slide, we use ancillary testing like visual fields to help evaluate these patients. And so these types of data were not available. And we also use a lot of imaging to evaluate patients in ophthalmology. And currently, All of Us does not include those types of imaging data. We found the user interface very easy to use, but the analysis still ultimately requires technical and programming expertise. And luckily, we had people with those skills on our team, but so it's not sort of a truly off-the-shelf type of service just yet. But overall, it was a great experience and a really great way to get to know the workbench and demonstrate that there are a lot of advantages to using a large national diverse cohort for this type of research. So thanks for letting me share a little bit about our demonstration project, and certainly happy to take questions if you have any.
Thanks, Sally. In the interest of time, and also because you covered so much already in terms of feedback gathered, I must just say we had another project that I won't get into the details now, but the next slide shows the same or a subset of the people also attending a culmination meeting at Vanderbilt in which the projects were discussed. So with that, the next slide just shows a few reminders of what Kelsey had already referred to in terms of privacy being a priority and what types of data transformations were necessary in order to protect that. And finally, the feedback gathered, as Sally pointed out, it was very easy to use. It's very exciting to have this resource available to all of us, and to be able to provide this type of constructive feedback so that it can only get better and better. And with that, I would like to stop. I do see there are questions for Sally directly, and some others that are more general. So while you're here, maybe you can answer the questions of how much of ophthalmology data are in the All of Us data.
Sure. Yeah, so as I mentioned, the ophthalmology data are pretty sparse. Let me just pull up, well, I can't share the slide, but I have what we did as part of the manuscript that we submitted describing the demonstration project is that I went through and looked at ophthalmic data coverage in All of Us. And as an example, if we looked at all participants with SNOMED codes related to glaucoma, that was about 6,500 patients or so. And the number with visual acuity screening, so a code for visual acuity observations, was only present in about 80 or so patients. And so that was about 1.3% of that cohort. Corneal pachymetry, which is corneal thickness, that's another thing that we evaluate a lot in glaucoma. That's about 0.5% of the entire cohort. Glaucoma is often associated with intraocular pressure management. And so as clinical ophthalmologists, intraocular pressure is something we monitor closely. And there are actually less than 20 participants who have that as a discrete LOINC observation in the data warehouse. So that equates to about 0.2% of the cohort with glaucoma. And so that just gives you an idea of, the sparse nature of ophthalmic observations. Luckily, our original predictive model was looking at the role of systemic medications and comorbidities. And so when we did the demonstration project that was validating that original model, we constrained ourselves to look at only those variables as well. And many of those elements are very well represented in All of Us, things like blood pressure, heart rate, and medications and so forth. So I think for projects looking specifically at ophthalmic data elements, that would be much more limited just because of the sparseness of the data there.
Yeah, so let me take the opportunity also to say that not only the number of participants is increasing, but also the richness of the data. And again, the data completeness task force and other groups are investigating areas that of improvement to enrich the data. With regards to the question related to if the demonstration projects will be information and papers will be available. The paper is still under submission, so I'm pretty sure as they become available, there will be a place in the link from the workbench that relates to them, and also a list of all the projects. So again, just to conclude, I think it's very important that this initial projects were launched to test the environment, and now the as it gets open to everyone, that people provide feedback. So a question regarding to feedback, I believe Kelsey showed some ways to give feedback from the interface already. And with that, again, I wanted to thank everyone. I apologize not having time to cover all the questions, but please do use the resource and provide feedback for the group. And thanks for the invitation to talk to you today.
Thanks, Lucilla. And we will continue answering questions in the Q&A chat box. But we're running a little bit tight on time. We have our next presenter is Jason Carnes. He will be presenting on racial, ethnic, and gender differences in obesity and body fat distribution, an All of Us Research Program demonstration project. So Dr. Jason Carnes is an assistant professor at the University of Arizona College of Pharmacy, Department of Pharmacy Practice and Science in Tucson, Arizona. Dr. Carnes' primary area of research interest is cardiovascular and immune pharmacogenomics, specifically investigating the utility of genetic polymorphisms and predict adverse drug reactions. He employs a translational approach to investigate pharmacogenomic associations using observational studies, functional genomic techniques, and eventually prospective clinical trials. Dr. Carnes directs the scientific programs core for the University of Arizona Banner All of Us Research Program site. He is active in multiple professional societies, including the American Society for Clinical Pharmacology and Therapeutics, the American Heart Association, and the American College of Clinical Pharmacy. He has received support for his research from the National Institutes of Health, the American Heart Association, and the American College of Clinical Pharmacy.
Okay, thanks so much, Raquel. So as a participant in the All of Us research program myself, I'm very happy to be here and be able to illustrate some of our experiences with respect to the use of the workbench, as well as some of the benefits and drawbacks of the All of Us resource. Just as a disclaimer, I'm not an expert in racial disparities and obesity, so I do have my collaborator, Dr. Jan Clementidis, who's the corresponding author on this work, on this call in case questions arise that require more specialized knowledge. So continuing on with our background, so I think everybody has a good appreciation for the health impacts of type 2 diabetes and cardiovascular disease. Just to introduce our project, we were trying to replicate a lot of the notable disparities in abdominal obesity and these diseases across different race and ethnic groups. So we used this first interim release of All of Us data to replicate previous findings with regard to some of these anthropomorphic as well as disease diagnostic metrics. And this really helped us to assess the suitability, the breadth, the depth, and the quality of the All of Us data. As you just saw in the last presentation, we were restricted to replicating previous findings as opposed to new discovery work. And we did focus on these racial and ethnic and gender disparities in body weight and body fat distribution, also evaluating levels of alanine aminotransferase or ALT which is a surrogate measure of hepatic steatosis. And then in terms of our methods, and I'd like to use this to sort of illustrate some points in terms of the resource. So all of us is really a patchwork of data sets. If that isn't clear by now, I'd like to reemphasize that point. So there are going to be varying numbers of total patients depending on what kind of data you want to acquire. So the participant provided information, which will be provided by the participants through surveys and other means, is going to be available on a different number of individuals than the EHR data is going to be available on. And so you end up with different n's, different numbers, different sample sizes, depending on what data you're collecting. So for instance, we used self-report to determine race, including non-Hispanic whites, non-Hispanic blacks, Asians, and Hispanics. As you can see here, there was a different question in terms of how an individual self-identified their race and ethnicity. And this was a single question which allowed you to select multiple answers. And you can see those listed here. But as I mentioned, the first release due to privacy methodology really condensed some of these categories. And it's called the data generalization steps to these non-Hispanic white, non-Hispanic black, Asian, and Hispanic categories. And so, for instance, all Americans reporting American Indian or Alaskan Native race and ethnicity had been removed by the time we got a hold of the data. So those individuals wouldn't be included. For biological sex, we did assess this at baseline and only used biological sex. I know Dr. Lund is speaking next, and he's probably going to tell us about how wrong we were by doing this and point out some of the limitations for exclusively using biological sex. But that is the approach we took, consistent with the prior literature that we were replicating. We used the standardized physical measurements that were obtained at baseline. So of course we had the option of using EHR measurements, but we concluded that, because we're using anthropomorphic measures that were acquired consistently across the All of Us research resource, we would have much more reliability if we use the physical measurements as acquired by all of us, as opposed to going into the EHR to either fill in holes or to see how these anthropomorphic measures changed over time. Now, ALT levels, on the other hand, we could only get from EHR records. And so you see the low ink code there that we used. Now, another thing about the laboratory values is that you look up any individual laboratory value, you're likely to get a number of different kinds of low ink codes and associated lab values. So there were multiple different kinds of ALT measurements available within the system, but we use the most common one essentially, and the one that sort of had a much greater preponderance than the other ones. So I think this one had about 40,000 instances or 40,000 individuals with data, whereas the next most had only about 2,000. And then in terms of the ALT measurements, of course, multiple ALT measurements might be available on each individual. So we use the most recent ALT measurement if there were multiple labs, but we did exclude ALT measurements that were taken prior to 2005. And that was a more or less arbitrary cutoff, but some ALT measurements had been taken 20 years prior. And so we expected that a time cutoff would increase the reliability of the data. Next slide. In terms of statistical analysis, again, I mentioned it was all registered tier, but the key with regard to the patchwork is we use the maximum number of individuals in each analysis that we could if they had the available data. And so I'll give a breakdown of that, I think, in the next slide. But of course, we had to do a little bit of data cleaning as well. So we had, unreasonable height, BMI, and BMI measurements, both at the low end and at the high end. And so we cleaned that with, again, relatively arbitrary, but what we consider biologically impossible measurements. And then, again, based on these privacy restrictions, we didn't report anyone or any individual or group of individuals below n equals 20. And we also excluded ALT outliers as well. Standard definitions of obesity, and we did use t-tests as well. We did non-parametric tests in terms of a sensitivity analysis, but as you'll see, there are enough patients in each of these categories where the parametric versus non-parametric testing didn't really make any difference at all. We compared our rates, or the ALU rates of age-adjusted obesity prevalence by race, ethnicity, and gender to the NHANES prevalences. And I have a figure on that a little bit later, as well as doing linear regressions to model BMI and ALT with the different variables, the different key variables that we collected, as well as gender by race interaction terms within those linear regressions. Again, this was all done in R in the Jupyter Notebook. Our code is available at this very long URL, but it is publicly available within the workbench. Next slide. So in terms of results, and I think this really illustrates the availability of the data, how the tiers of individuals keeps decreasing depending on the data that we want to use per analysis. So in our initial data using the registered tier, there are over 242,000 participants available, but only 224,000 of those had the necessary PPI-derived data on race and ethnicity and gender after those data generalizations were implemented. And then we have a decreasing N based on those used in the analysis of anthropomorphic measurements after all the data cleaning that we did. And then finally, with the EHR, in terms of those who had a usable ALT measurement that wasn't an outlier and that was acquired after 2005, so the numbers dwindle in terms of, it's still quite large at 46,000, but that was the numbers that we had. And that, I think, really illustrates the patchwork data set. Not everybody in the data set has EMR data and PPI data and anthropomorphic data available. In terms of results, the body mass index was highest among non-Hispanic Black women. And you see the mean there at 33.3, and it was lowest among Asian women. The gender difference, as we have seen in the prior literature, was really most pronounced among non-Hispanic Blacks. So the non-Hispanic Black women had a much higher BMI than non-Hispanic Black men. That is well replicated in the current literature. And you can see here the p-value, you may recognize that. That's the lowest p-value that R can spit out, to my knowledge. So we're working with big numbers in terms of N's, and that, of course, leads to what some might consider an overpowered analysis, but certainly significantly different with the number of individuals that we have. We saw a similar effect among Hispanics, but this gender discrepancy was almost absent in non-Hispanic white individuals. When we're using linear regressions, we saw an increased BMI with age. You can see the betas and p-values here in women versus men, in non-Hispanic Blacks versus non-Hispanic whites, and in Hispanics versus non-Hispanic whites. So using terms that included both the race ethnicity variable as well as the gender variable that we used in the age, everything pretty much had an influence. And naturally, we also, when we use these interaction terms, we saw significant interactions in virtually every analysis we had based on the numbers. So we did find significant interaction terms for each of the race groups. And so here's a representation of some of the raw data that we have. You can see here, I guess the first thing to focus on would be the N's. We have a different N on the left side, which is really the total numbers. And you can get a pretty good idea of the race distribution, at least in the tier that we acquired data on. And then there's an N to the right where you can see, naturally the ALT had dramatically different N's than our anthropomorphic analysis are. So we felt obligated to report those differently on the right side of this table. But if you advance the slide, you can see the highlighted BMI in non-Hispanic Black women and non-Hispanic Black men, and the discrepancy there that was really one of the hallmarks of the prior literature and what we saw repeatedly throughout our analysis. And that is, of course, backed up by the prevalences of obesity that are right next to there, with there being a highly discrepant obesity prevalence between non-Hispanic Black women and non-Hispanic Black men, again, which is replicating what the previous literature sees quite strongly. And the next slide. And here's our comparison to the NHANES data. You can see that we used two comparisons. One was the prior 2015-16 NHANES, and then the 17-18 NHANES, which had recently released before we published. And as you look along these, the bar chart, you can see that just in general, broadly, the all of us obesity prevalences are a little bit lower than the most recent NHANES estimates. But that doesn't ring true for every group. For instance, non-Hispanic Black women and Hispanic women have higher obesity prevalences than all of us. And although it's sort of a tenuous interpretation, one of the things we wanted to assess while we were doing this project was, is there a healthy participant bias within all of us? In other words, are we recruiting an overrepresentation of healthy individuals within the resource, similar to how UK Biobank works? There's a well-known healthy participant bias there. And I think based on our data, based on what we see here, the conclusions is that it's probably, if it exists, it's probably subtle. We did see some evidence of a healthy participant bias within all of us, based on these lower obesity prevalence estimates, but it wasn't consistent across all groups. So we considered that to be fairly subtle in terms of the healthy participant bias. And the next slide. And in terms of our ALT analysis, so we also replicated a number of observations that have been seen in the previous literature. Men did exhibit higher levels of ALT than women. And then in both men and women, non-Hispanic Blacks exhibited lower levels of ALT compared to all the other race groups. You can see our betas and p-values in terms of the linear regression modeling as ALT decreased with increasing age in women versus men and in non-Hispanic Blacks versus non-Hispanic whites. ALT also increased with increasing BMI. And then again, we saw significant race ethnicity by gender interaction terms observed for all race and ethnic groups. And so in terms of our conclusions, so again, we replicate these known racial and ethnic and gender disparities related to obesity. These are very pronounced differences, and even more so in non-Hispanic Blacks. These are known and likely due to genetic factors as well as socioeconomic factors and sociocultural factors as well. Non-Hispanic Black women, however, did have a lower ALT despite that higher BMI, suggesting a quote-unquote metabolically healthy obesity phenotype that has been seen in the prior literature before, and is a concept that has been brought forth. A minimization of hepatic steatosis, even though a higher BMI is present, and those are usually correlated variables. Our data does suggest that, at least with respect to obesity, our averages are representative of national averages. We did observe some evidence of a subtle healthy participant bias based on our age-adjusted obesity prevalence rates. These were generally across all our race and ethnic groups, but again, not consistently across all the groups that we evaluated. And then we do think that, based on the sample size, the massive number of individuals, and this was, again, an interim release of the data, the incredible diversity here, and the availability of these multiple types of data that can be combined, although that comes with limitations, It also comes with major benefits, typically that the sample sizes that you can get and the depth of data, the phenotype dense data that we have available here. And that's before the whole genome sequencing data and the array data are available, which is going to make the resource even more rich, as well as something that's been mentioned before, the ability to recontact patients and to get phenotype or system-specific data that has been collected prospectively for that particular purpose should be very powerful as we move forward. Some notable limitations to our approach. We did exclude based on pregnancy, we did not make any exclusions based on pregnancy. We didn't account for comorbid disease or drug treatments for obesity. And then, of course, this patchwork of data sets creating an inconsistency in the ends that we have for the data. And to just revisit some of these considerations around the workbench, we felt that cohort builder, the saving derived data sets was initially challenging, but it works quite well now. So that speaks to the DRC's iterative approach in taking feedback from the demonstration project groups and really working to make that to make it more implementable within the resource. The link between the data sets and the contents of the data browser was quite good. The Jupyter Notebook system, I think is becoming more popular and really forces us to do more rigorous and reproducible bioinformatic research that's trackable and improves the research methodology overall. We did see some issues in terms of data consistency, especially if you're coming from multiple sources in terms of EHR and anthropomorphic measures from the research program. So as much cross-checking and plotting of the data, looking at outliers, ensuring data quality across this breadth of information and participants will be important. The data are pretty clean and standardized using those the terminologies that have been previously discussed. One thing that our analysts said that it would be nice to see more easily query and analyze program data versus the EHR data. So the connectivity, and I think the merging was mentioned before with respect to the workbench. And again, the cohort builder, the concept sheets, they mimic the research process, and this was definitely a strength. And the visualizations were helpful, but it would be nice to have more quality figures for representation. So next slide. These are considerations that I'll go through quickly with respect to EHR data, mostly, not specific to all of us. And I've been through a lot of these. You have to, of course, consider timestamps, when your lab is being done respective to whatever phenotype you're defining and at which point you're defining it, this patchwork of data sets. Also, one critical issue that a lot of our demonstration project teams had was the denominator, right? So there's this denominator problem. If you're creating a cohort of cases and controls, then you have to define them similarly because the absence of your criteria for defining cases and controls may not be consistent across both groups. And that's definitely something that we saw with a lot of demonstration project groups. So if you're looking at prevalence estimates, then who doesn't have the disease is going to be important in terms of the overall cohort. And again, I mentioned healthy participant bias. So next slide. And I think this is just acknowledgements. Of course, want to acknowledge all the funding and of course all the participants. And with that, I'd be happy to take questions.
Great. It does look like we have a couple of questions here. So ATL has a pronounced diurnal cycle. Does the EHR document the time of day?
For ALT specifically, Jan, do you, I believe it does. I believe there's a timestamp associated with all of the labs, but I didn't work as closely with the data. Jan, can you answer that?
That's a very good question. I am not sure. We need to check on that, but that's a great question.
Yeah, so that's maybe something we should consider in updating the analysis, is diurnal variation in ALT.
Okay, and I don't see any other questions. If you have any, please type them in the Q&A. Okay, so I think we can go on to our next demonstration project if nobody has any comments or any more questions. Actually, we have a new one. Are you collecting cortisol? If so, same issue about time of day it was collected.
So we hadn't collected cortisol. Certainly there are going to be a lot more variables that influence the phenotypes that we collected. We sort of looked at this as a more the low-hanging fruit in terms of anthropomorphic measurements and the EHR measurements. That is an excellent point. We could also look at cortisol and also apply the diurnal variation and try to tease out the effect of the time of day. And so we haven't looked at it at this point. My only worry would be the increasingly low numbers that you would get, but because of the large starting in, there's got to be a number of individuals that have all the data that we collected plus the cortisol data as well within a reasonable time frame from when we're defining our phenotype. But that's another great suggestion. Thank you.
Okay, great. Well, thank you so much, Jason, for your presentation and for sharing with us. We're really glad to have you here. Next, we have Mitchell Lunn. He will be presenting health outcomes of sexual and gender minority people, data from the All of Us Research Program. Dr. Mitchell Lunn is an assistant professor in the Division of Nephrology of the Department of Medicine at Stanford University School of Medicine. He is the co-director of the PRIDE study, an online national longitudinal cohort study of over 20,000 LGBTQ participants, and the co-director of PrideNet, a national LGBTQ community engagement network. Dr. Lunn has a special interest in using technologies to support participant recruitment, cohort management, and retention and results dissemination. Clinically, Dr. Lunn provides adult primary care in Stanford LGBTQ health program and attends on the inpatient nephrology consultation service at Stanford Hospital. Mitch earned his Bachelor of Science degree with highest thesis honors from Tufts University in 2004. His Doctor of Medicine degree from Stanford University School of Medicine in 2010, and his Master's in Advanced Study degrees in Clinical Research from the University of California, San Francisco in 2017. He completed internal medicine internship and residency training at Brigham and Women's Hospital in 2013 and nephrology fellowship at UCSF in 2016.
Great, thanks Raquel, and thanks to the organizers for inviting me. It's really a pleasure to be here and happy to talk to you about an underrepresented population in biomedical research, which are sexual and gender minority people or LGBTQ plus people. And if we can go to the next slide, please. And so I wanted to start by telling you a little bit about our group at Stanford, which goes by the name of PrideNet. We're a national community engagement network, and we're also a national community engagement partner for the All of Us Research Program. Our work focuses primarily on engaging sexual and gender minority people to participate in the All of Us Research Program and educating them about what that means. We also do some work inside the consortium, inside the All of Us research program to train frontline staff, to train NIH staff, to train others, to be really welcoming and affirming in their interactions with LGBTQ plus people, so that it's really a positive experience for SGM people to participate in the All of Us research program. And then we also engage external researchers, many of the folks on this call, and looking through the participant list, I see some colleagues and friends to really participate as researchers in using the All of Us research program data for their own LGBT plus related research, as well as other research endeavors, especially including those related to health disparities. Listed here are members of our team specifically on this project, and a big thanks to all of them. A big thanks to the demonstration project subcommittee, and of course to all of us participants that made this possible. Next slide, please. And so for folks who may be less familiar with the LGBTQ plus population from a health perspective, the term that is now commonly used in academic medicine and commonly used in the NIH, and I see folks from the Sexual and Gender Minority Research Office on this webinar as well, is the term SGM, or sexual and gender minority, and this really refers to folks who are not heterosexual, so people who are not straight, and then people who are not cisgender. So cisgender refers to having a gender identity that is most consistent with the sex that you were assigned at birth. So for example, most of us in the world are cisgender. I'm a cisgender man. I was assigned male sex at birth, and I identify as a man today. And so sexual and gender minority people is a kind of an umbrella term to refer to people who are not straight or people who are not cisgender. And we know from some research out there, and these are typically smaller studies done with lower sample size, smaller sample sizes, and in a defined geographic region, that there's increased rates of, for example, tobacco use, some cancers, HIV infection, depression, anxiety, and other things that are thought to be related to a concept of minority stress. So living in a world that is stigmatizing and discriminating results in both mental health and physical health downstream effects. And I think what makes all of us really exciting is that, for the first time from the very beginning in a federal study, has sexual orientation and gender identity been collected on all 1 million participants, or on all participants that have enrolled so far, with plans to collect it from all 1 million folks. And so this combined with the program's additional commitment to having large numbers of folks from underrepresented communities, that the diversity of the data set, not just from a sexual and gender minority perspective, but also, say, for example, from a racial and ethnic perspective, from a socioeconomic perspective, all really makes this an excellent resource for studying SGM health disparities in a really diverse population, diverse more than just sexual orientation and gender identity. Next slide, please. And so the question that we sought to answer here was, what are some of the health conditions or behaviors of SGM people compared to non-SGM people in the All of Us research program? And really, as you've heard throughout the presentations today, looking at previously reported health disparities, are we able to reproduce some of those or not? And also we wanted to really look at the diversity of just the SGM subset of participants in All of Us and compare it to the non-SGM side of things. And so next slide, please. And I'd like to spend a little bit of time on this slide, which really focuses on the basics. PPI, or participant provided information module, or a survey called the basics, that all participants in All of Us are invited to take. This is where sexual orientation, gender identity, and sex assigned at birth are collected. As you've heard earlier about data generalizations in the registered tier, which is the current tier that's publicly available, and how this can affect analyses. And I think that's an important aspect to talk about here. So, well, sex assigned at birth has been generalized to show either male or female. So if you were selected intersex or selected something else, those options are not, that granular level of data is not available to protect participant privacy. And similarly, currently in the registered tier, any sexual orientation other than straight is generalized into a category that's called non-straight or unknown. And so if you selected bisexual or lesbian or pansexual, those are kind of all collapsed down into a single category. And similarly for gender identity, if the gender identity selected was something other than man or woman, for example, transgender, genderqueer, gender variant, gender fluid, those are all collapsed down into a different category. And so we ended up basically creating 4 different sexual and gender minority subgroups, which are shown here based on the generalized data that's available. Of course, there are many limitations with generalized data, and one of course the future directions here will be to use the non-generalized data when the controlled tier is released in 2021. I think this is an important part to just highlight for people who are not doing research in sexual and gender minority people, that this really is, especially the difference between sex and gender, is a really important thing for everybody to consider about when they're doing their analyses with the All of Us research program data. So if you're interested, for example, in looking at anemia in quote unquote women, you need to, I think, really understand what you're wanting to look at in terms of when you say women. Are you looking to as in people who are assigned female sex at birth and currently identify as a woman, or are you looking at only people whose current gender identity is woman, which may include people who were assigned male sex at birth? And so for folks who are not necessarily used to thinking about the difference between sex and gender, this is, I think, an important time to do so as you're working with the All of Us data, because it will affect the cohort that the subset of the cohort that you're getting for analysis. Our group created a very brief handout that has been basically an educational material for research and for researchers to really understand this difference, and is, I think, an important thing for folks to consider, especially if you're trying to reproduce work that was done previously. Typically, gender, this papers often conflate sex and gender and refer to gender in terms like male and female, when really they were talking about what sex they were assigned at birth. So just important things to consider as you're planning your analyses, because these data are collected on all participants and all of us. It's an important thing as you're defining your cohort to do your analyses. So we looked at each of these 4 groups here, and then of course we had two non-SGM groups, which were cisgender straight men and cisgender straight women. And we looked at really a whole slew of demographics, as you can see here, age, race, ethnicity, etc., to look at the differences between the 2 different groups, and characterize those, not using a chi-square test, which a lot of people use. We instead use usually the absolute standardized difference statistic. And so we can go to the next slide, which just highlights some of these differences, which some of the things were a little bit surprising. So you can see that a ASD of around 0.2 is a small difference, 0.5 is a moderate difference, and 0.8 is a large difference. You can think of this similar to a Cohen's D for folks who are familiar with that statistic. But not surprisingly, the SGM folks were, compared to the non-SGM folks, were younger in age, with kind of a small to moderate effect. We were a little bit surprised to see that this was actually slightly more diverse in terms of race and ethnicity in the SGM group than the non-SGM group. Typically in most SGM research studies, they're usually rather white predominant. And we had a good distribution across the country of SGM people. Of course, a lower education and lower income, which likely really is related to the decreased age, and then a slightly higher increased amount of uninsurance or not being insured. But overall, found these groups to be rather, kind of representative of the rest of the AOU cohort, and there was not large differences amongst the SGM side. Next slide, please. And so we wanted to look at these health conditions, most of which, all except for a body mass index greater than 25, all of them except that one were done using EHR data that was available, and the body mass index was done from the program obtained measurements in the physical measurements and biospecimens component of the program. So when people go in to get their physical measurements obtained, we use the program obtained measurements rather than measurements that may be existing inside the electronic health system. And then next slide, please, just to discuss a little bit about how so we identified really SNOMED codes that were specific for each of these different conditions. And required that at least there be one SNOMED code on 2 different dates. So you can imagine that sometimes, as a provider, I erroneously code things that may not be a true diagnosis, but something that I'm suspicious for, and so that may be a little bit erroneous. And so instead, having a SNOMED code, say for anxiety on multiple dates, was required for inclusion as somebody having anxiety. We ran logistic regression models, adjusted for age, race, ethnicity, and income, and used an appropriate a non-SGM reference group. So, for example, folks who were, say, cisgender sexual minority women. So this may be, one example of somebody who that would be somebody who was assigned female sex at birth, now still identifies as a woman, and has a bisexual sexual orientation. We compared those folks in that group to cisgender straight women. So really, comparing it to their non-SGM counterpart. However, due to the data generalizations, gender minority groups contained folks who were transgender and identified on the gender the man-woman binary, and as well as folks who were gender expansive, who may not identify with a particular gender or identify with a gender outside the male-female, man-woman binary. And so for all the gender minority groups, we actually ended up comparing them both to their straight cisgender men and to straight cisgender women, and making those comparisons, given that it was unclear if there was a quote unquote correct reference group to use. Next slide, please. So just to review some of the data, I'm not going to go through them all because there's many of them, but really this is showing some of the differences and a lot of things that were not necessarily surprising. So this is among cisgender sexual minority men, so people who were assigned male sex at birth identify as a man today and have a sexual orientation other than straight. That there was obviously a very kind of striking odds of HIV infection, as well as some other physical and mental health disorders that were, I think, not necessarily surprising to us, and it was great to see that these data were replicated. However, if you go to the next slide, you'll see that I just picked out 2 of these compared to some data that exists in the literature. For anxiety, for example, the previous literature estimate of a kind of different population, much smaller in sample size, had an odds ratio significantly higher for anxiety, with a 95% confidence interval that actually doesn't overlap at all with what we found in all of us. And so I think some of these data were reproducible, other data were not, and I think that's not necessarily a flaw of all of us per se, but is more likely a flaw from using smaller sample sizes and how the other samples were collected, which is oftentimes in kind of these bar or social settings, rather than in electronic health records or in any other really epidemiologic way with a reasonable sampling frame, because we don't have great sampling frames for sexual and gender minority people across the country, because sexual orientation and gender identity are not included in the US Census. So we actually don't know where LGBT people live in any kind of reasonable way. Other existing items in the literature were rather clear. Things like tobacco use, mimicked previous data from the CDC, from an MMWR, and so I think that those really, were reproducible, which I think was, bringing out, good confidence in the diversity and depth of the data. Next slide, please. And so just to give an example of comparing to having for a gender minority folks and comparing it to two different reference groups. So this is comparing folks who were assigned female at birth, have a gender identity different than woman, something other than woman, and then comparing this to straight cisgender women, a variety of physical health and mental health differences here. However, when you compare this group on the next slide to straight cisgender men, you can see that there are different ratios. And so next slide, please. Here, compared to cisgender men, gender minority people assigned female sex at birth actually have, quite strong rates of anxiety, asthma, depression, and for the most, and to a lesser extent, having a BMI greater than 25. So these aspects, I think, were to be honest, not necessarily known, and that's because there's not a lot of data among gender minority people in the literature, period. And so next slide, please. Just to wrap up with a few slides here, I wanted to chat a little bit about some conclusions, and that was that we did, the previously reported health disparities were often replicated, but not always. We did find replicated or odds ratios that were not what was seen previously in the literature. We also frankly found some new associations because they hadn't really been studied before, because they just did not have large numbers of gender minority people to do this type of work, which I think is a testament to the diversity and broadness of the AOU data set. And then thankfully there's very small differences in the demographics between the sexual and gender minority people and the non-SGM people, so which would fit with what we would expect in reaching a diversity, really an intersectional diversity, not just of sexual orientation and gender identity. On the next slide, some limitations are, of course, the big one, the kind of elephant in the room for this research project, for those of us that are LGBT health researchers, is the data generalizations, and kind of the way that we don't like that that various sexual orientations are collapsed into a single item, and similarly, various gender identities are collapsed down into a single bin. So we're really looking forward to the control tier data release later in 2021 for where those generalizations will be, will be removed. Of course, this is a volunteer sample, so it's not a probability sample or any random sampling, and it's obviously observational data. I think what we heard a little bit from Jason is just to be aware of missing data, or how that denominator changes as you move smaller and smaller down the funnel. And while there's the denominator changing, there's also varying amounts of missing data at various stages of that process. And that can be missing data both from surveys as well as from electronic health record data for particular participants. And so it's really just important to be cognizant of that, to look at it, to be aware of various missing data analysis techniques and use them appropriately. And so the next slide, some implications for this is that we think that this project will add to the body of literature characterizing SGM health disparities. It's really a prioritization for research areas that can help prioritize areas that need additional research. And I think it really demonstrates the fact that the All of Us research program is going to be, I think, a game changer, or revolutionize health disparities research by the commitment to having an extraordinary percentage of folks who are underrepresented in biomedical research. Next slide, please. And so just some lessons learned, I think, some valuable skills that are important in this work is to understand the OMOP data model as best you can. There's lots of resources online and lots of help within the researcher workbench as well. Knowing SQL is helpful to really query the database in an appropriate way. And then, as I mentioned, some approaches to deal with missing data, even just the appropriate ways to do the analysis. We had great interactions with support, including a kickoff meeting for this demonstration. You've seen some photos and other things. We've done various help requests and responses that way and by email. And then just some things to be aware, which I think you've heard from other folks today, is just the details of how the All of Us data is curated, how it comes together, that the EHR data coverage varies from data type to data type necessarily. There may be some non-obvious data limitations, like the data generalizations especially, or making sure that you're aware of which constructs are being measured. So the Survey Explorer, which is on the workbench, is very helpful to take a look at the actual survey questions and the answers that were there. And then just be aware that you may run into some computing limitations. So just be careful on which queries you run. We've, you can accidentally run a query that returns every single data element in the in the entire database, which takes some time and can be problematic. So, and then again, looking forward to doing these analyses in different ways once the control tier is available. So with that, sorry for going so long. Thanks to everybody, and happy to take questions if there's time. Thank you.
Thank you so much for your talk. It was really exciting to hear about an underserved in research population. We have a couple of questions for you that we're gonna try to get to. So there's a bunch of interest in the handout on sex versus gender that you described in your talk. Could you send that out? If you can send that to us at All of Us, we can send it out to all of the participants so that they can have access to that as a resource.
Sure, that would be great. I think it will also be making its way to the researcher workbench as well, so people will be having it there. But yes, we can get it definitely out to all the people attending this. That would be great.
Excellent, we appreciate it. So one question came in from Carl Streed. Why compare gender minorities to straight cis folks rather than to all cis folks, unless you're accounting for sexual identity in both gender minority and cis groups?
Yeah, and I think Carl, we played. Nice to hear from you, by the way. We played with this idea also as to just binning all the straight cisgender people together as one reference group. We did want to, I think, not lose potential biological differences from sex assigned that may be implicated by sex assigned at birth. I think the other limitations here, of course, are that for folks who are on gender affirming hormones, for example, we don't necessarily know that status, or that status was not a part of this. So instead, we made an assumption that most of the gender minority folks in the All of Us research program were not taking gender affirming hormones and wanted to keep thus some analysis comparing to both sex assigned at birth. In the future, a great analysis would be to also kind of control for gender affirming hormone status and folks who may be taking hormones as well. Thanks for your question.
Great, and I think the last question, because we're running a little behind schedule, is thank you so much. There is a lack of gynecological data about uterine fibroids, endometriosis, prevalence, et cetera, among sexual and gender minority populations. Is that something that could be, or was of interest to be generated from this?
So we did not look at that. I think it most definitely is a possibility because you could look at EHR codes for fibroids, for endometriosis, and then also look at that by sexual orientation and gender identity groups, and creating those groups either using the generalized data now, as we've done, or using the more granular data in the control when the control tier happens. So I think that is a great project. Happy to chat with you about it. Would definitely be an easy one to do using the data here. If we're looking at EHR codes, if you're looking at other things like, you know, imaging or procedures, those options are available, but are a little different. And you know, that analysis is a little different.
Great. Thank you so much for all your time. And I think that'll wrap us up.
Great.
So with that, I'd like to say hello and thank you. This is just your first time pretty much hearing my voice, but I've been communicating with over hundreds of you on our chats. So thank you so much for your involvement and feedback throughout this process. Next slide, please. I wanted to give a huge thank you to everybody who is involved in the Research OnRamp development. The team that organized this event is included here on the right, and photos of us are on the left. And I wanted to say thank you to everybody who came here today. Next slide. I would encourage everybody here to share information about the All of Us Researcher Workbench amongst your collaborative teams, with your trainees or students, with anybody you think would be interested. This is a resource designed by all of us, for all of us, with all of us in it. And so the recording will be available soon, and we will send out an email, and you are welcome to share that. Finally, we encourage everybody here to join our research community. We would love for you to come in and kick the tires on our data, try out new analyses or techniques, or really dive deep into whatever field you find compelling. So with that, next slide. I would encourage all of those who have signed up for the onboarding walkthrough to return at 2:25 to their particular breakout session. You should have received an email that has a link to that session. Everyone else, I hope you have a wonderful day, and I hope we see you on the workbench. Thank you so much. Bye.